Is Evo Devo evidence for evolution? No, busted. I'm here with Dr. Cornelius Hunter. He's a biophysicist and he has studied the history and philosophy behind evolution. And he's going to be talking about how Evo Devo has not turned out to be this great thing that it was promoted to be. And so we're going to look at what he has to say about this. Thanks for being here, Dr. Hunter.
Thank you, Rebecca. I'm delighted. Thank you for inviting me on. And if you could bring up the slides, I wanted to just remind people where we are. So we're going to talk about embryology today. This is the fourth in a series of 6. So the first one was nested hierarchy, then we had homology, then we had small scale evolution, today embryology. And in each one we had a theme, a sequence. We've seen that there are very powerful evidences that evolution is set forth in these categories for evolution, making evolution kind of a slam dunk. But then there's actually major problems with these evidences, and it really turns sour, and you get the impression that this thing is really falling apart. So what's going on here? Because despite these problems, evolutionists continue with their claims. They don't drop these claims. They don't lose a beat. So that's kind of strange. Well, the third step, the persistence of the evidence. So we'll see that again today. We'll get to that in just a second. So embryology. So I want to talk a little bit about where it starts, because you have to talk about where it starts back in well, I don't know if this is exactly where it starts, but the 19th century, before Darwin and into Darwin's time, where embryology was in its infancy. And there were several arguments that Darwin made for why embryology is a key, powerful evidence for his theory. So Darwin, and after Darwin, embryology was held to be this, right up there with the other major evidences for evolution. And Darwin had some observables. So he had some inputs, some things that he was basing his reasoning on. And the one that I want you to remember, if you remember nothing else, it's resemblances. So there are powerful, very interesting resemblances between the embryos of different species, a fish, amphibian, mammal, bird, so forth. You look at their embryos and they look similar at a particular stage or certain stages of their embryological development. So they obviously don't look similar toward the end of that development, where they look more or less like how they should as a baby. But at an earlier stages, they look a lot alike. So that was his number one most important observation that he founded his conclusions on. There's these strong resemblances. So remember that. That is the most important one. He had some other ones that he kicked around. Like one of them was that earlier naturalists before Darwin, earlier in the 19th century, had said that embryology was a really important, the embryonic features were really important for classification. If you want to classify the species, the embryonic features work really well. So he kind of leveraged that one a little bit. Another one that he said was that the embryonic features are not necessary. There's probably not a real strong reason for most of the embryonic features. They could have been different. And he had reasonings for that, but he claimed that they weren't adaptive. That's the term of art. Instead, they were kind of neutral. It could have been different. Most of the embryonic features could have been different. So he had that one. So he had several of these. He would call these, fundamental observations or very strong conclusions based on observations. Then out of that, he made several arguments, and that's what I want to tell you about, these conclusions that he made that he thought were slam dunk, almost necessary, almost like a logical proof. These were, we may safely assume, we may safely infer. He used that sort of language for these conclusions. And one of these conclusions was that species have a less modified form, and the embryo, an organism's embryo represents that less modified form. Now you say, well, okay, that's interesting. What's a less modified form? I've never heard of that. Well, that's a major part of the book, of Darwin's argument. He's been cooking this. He's building this argument since the beginning of the book. Since the beginning of the book. By the way, the embryology arguments in chapter come late in the book. This is chapter 13 of the first edition, 14 of the sixth edition. So it's late in the book. He finally gets to embryology. So he's been kind of behind the scenes working up this concept that there's this less modified form. What is this less modified form? You get to embryology, you get to chapter 13, especially page 449. A lot of things come together on page 449 of the first edition. It's kind of unusual because Darwin's book is long and meandering, and he takes a long time to get to his point. He has long passages. So you usually have to read many pages and put things together. But in this case, at page 449, a lot of things come together, a lot of his major claims. And this first one, there's this less modified form, and the embryo reveals that. If you look at the embryo, you see it's less modified form. Well, what he's getting at is the earlier evolutionary stages. So that's what he means by a less modified form. But he wants to pass off this idea of a less modified form as a real slam dunk, like this is a necessary conclusion. So if you agree that there's a less modified form, that's the earlier evolutionary history of this species, then you've really bought off on evolution. So he wants to make this case. And the embryo represents that, or it reveals this less modified form. And what a tantalizing concept. If you're an evolutionist, you mean I can look at the embryo and actually get a glimpse of what's gone on in the past? That was, very tantalizing, right? So another one of his inference or conclusions, almost necessary conclusions that he's arguing for, is the embryo reveals the structure of its progenitor. That's a similar idea, right? These are very similar to the previous idea. It reveals the structure of its progenitor. If two animal species pass through the same similar embryonic stage, then we may feel assured that they have both descended from the same parent. They have common ancestor. That's the third one. So he's building all on this page 449, by the way. Thus, community of embryonic structure reveals community of descent. I like the turn of phrase. You have to be a little bit artsy with your writing to compel people. So he comes to that. Community of embryonic structure reveals community of descent. In other words, if we see different species that are very different in their adult stage, we see similarities in the embryonic stage. They share a common ancestor. That turned out
makes sense to me, right? Why would it be otherwise? What could possibly be the other explanation?
We'll get to that, but it's a very powerful argument. It's a very tantalizing. It captures the imagination. Yeah. Why would it be otherwise? We can clearly see why ancient and extinct forms of life should resemble the embryos of their descendants. Okay, what he just said was the extinct forms resemble the embryos of their descendants. So he's kind of going the other opposite direction. Extinct species resemble the embryos of their descendants. So if you look at the embryos, you get a glimpse of the evolutionary history of a species. So that is really building up to this idea of you can look at the embryo and get evolutionary history out of it. That set the stage for another very interesting character. Only a few short years after Darwin, like 5 years or so, into the mid-1860s, Ernst Haeckel, a German naturalist, Ernst Haeckel, and he got that idea in his head and thought, this is great. And so he came up with a pithy phrase. He was a very creative guy. He came up with these pithy phrases, and he came up with a pithy phrase to capture this idea, how powerful this was. Ontogeny recapitulates phylogeny. Ontogeny recapitulates phylogeny. So what does that mean? Ontogeny is being, the creation of the being. That's embryonic development. So ontogeny, or embryonic development, recapitulates phylogeny. What's phylogeny? That's the evolutionary tree, evolutionary history, the lineage, the past lineage. So ontogeny recapitulates phylogeny. You look at the embryo, you can see the past. Very powerful concept, very attractive concept for evolutionists. Haeckel then made drawings of embryos from different species and showed their incredible similarities and how they lined up in these, and how the evolutionary history was kind of revealed. So this was, again, the mid 1860s. Darwin, in his 6th edition, which is the kind of the edition that had more changes in it, where he incorporated, he took into account, criticisms, but also positive evidences like this. So in the 6th edition, Darwin constructively approved of Haeckel. And, well, there's a big story that goes along with Haeckel's drawings, and that's not the focus of this podcast. It's a little bit controversial. I'll just briefly mention that it turned out to be, it was accused of fraud. It got picked up by a later evolutionist in 1892. So 1860s, 1892, George Romanes, he wrote a book, kind of a textbook on evolution, and incorporated Haeckel's drawings. He modified them a little bit, but they're basically based on Haeckel's drawings. And those were then used in textbooks throughout the 20th century. And you can still find them today. I just found one, this week online easily. I wasn't even looking for it. And there was Haeckel's drawings presented as evidence. But it was actually George Romanes' drawings, but presented as Haeckel's drawings as strong, powerful evidence from embryology for evolution. So it has persisted a long time. But in 1997, about 30 years ago or so, an evolutionist named Michael Richardson scathingly criticized these drawings, these sketches from Haeckel, as fraudulent, that he had just exaggerated wildly and come up with these drawings. About a decade later, there was some pushback on that. A historian named Robert Richards wrote a paper, "Fraud Not Proven," where he argued, no, wait a minute, let's ease off here. It's not so bad. So, one way or another, but I think it's clear that these drawings were exaggerated. There really were a partisan attempt to beef up the theory and kind of manipulated the evidence. I think that's clear.
Yeah. Now I'm still showing the slide for embryology. Are you showing the slide for drawings?
I am. I'm not sure.
Okay.
I don't want to show any slides because I want to talk a little bit.
Got it.
Thank you.
Okay.
Thank you, though. So I'm just giving this history that, yeah, there's all this contention. And if you thought that's where I was going to go, I'm not. That's not the point of this, but I need to clear that out. That what came out of that, though, was evolutionists did, in the 20th century on up to today, strongly view embryology as this powerful evidence, even though ontogeny recapitulates phylogeny has been kind of viewed as, oh, that's really not legit. It's not that literal. You can't literally go to an embryo and just, aha, there's its evolutionary history. But don't throw the baby out with the bathwater, says Jerry Cohen. There's still a lot you can glean from the embryo and the structure of the embryo, and you can still find hints and certain stages of its evolutionary history right there. So it's a valid concept. You just have to be careful with it. And so you see that throughout the text throughout the 20th century. It continues to be a very powerful argument. So don't worry about the controversy and Haeckel and all those drawings and all that stuff. My point is, embryology became very powerful, but in ways that were kind of subjective. One of I guess the poster child of that claim that you can see historical past. So this idea, you can think of this idea as evolutionary history kind of adds layers to the embryo. So over time, if you look at the state, the embryo takes months to develop. So if you look at the stages as the embryo is developing, there's kind of additional, metaphorically I'm speaking, layers laid onto it. It's not literal at all. That's not how embryology works. But you can think of it as evolutionary history laid those additional phases or additional stages on. So as you look back to earlier embryonic stages, you're seeing earlier evolutionary history. Probably the poster child for this is the gills. So fish, we all know, have gills. And what are gills for? Gills are a very sophisticated breathing system where water is brought through this channel, and the fish extracts the oxygen in the water. And it's very sophisticated. And when you look at the fish embryo, you can see the structure there that's going to become the gills. They're called pharyngeal pouches and so forth. So when you look at the human embryo, it has a very similar thing. And so you can imagine what people like Haeckel and George Romanes and later 20th century evolutionists did with that. They said, oh, there you are right there. That's an example of ontogeny recapitulates phylogeny. You can see the common ancestor that fish and humans have, mammals have, right there. The mammalian embryos show these gills, these gill systems. Well, so that has been a very attractive claim, and that's a very powerful evidence for the evolution. So I'm talking, this first section, I'm presenting the evidence for evolution. And you can see this one is a little bit tricky. It's a little trickier than the others. Usually evidences and arguments for evolution are somewhat straightforward, and then they have problems. This one is a little more subjective and a little trickier, and you have to kind of understand this history. But the point is, you look at the embryos and you see these hints of evolutionary history, and the gills, the gill slits of the human embryo are just really powerful. If fish have gills and the human embryo has gills or gill slits or a gill system or something like that, that's pretty interesting. That's a pretty interesting observable that evolutionists certainly have made a lot of hay off of, and they're still today making that claim. So this is not something that is back with George Romanes or Haeckel or Darwin. This is a contemporary claim of evolutionists that there's these gill slits, and in the human embryo, and that's very powerful evidence for evolution and common descent. Now, was there anything else that I wanted to say about the evidence? I have a bunch of slides I'm going to flip through, and I will show what I've been talking about, and you'll see a little bit more detail here. Powerful evidence. So I've got the summaries there. So here's Michael Ruse claiming that embryology fits in right with all the other categories of strong evidence. But why? So I'm going to flip forward to page 4. Okay, so here's a sketch of those embryos, an illustration of those embryos. This is from George Romanes, but it's based on Ernst Haeckel's work. So it gives you a kind of idea. So on the far right here, you have a column of 3 embryos. That represents the human, this different stages of the human embryo. And then next to it, you've got the rabbit, and then the calf and the hog and so forth. And if you go to the top row, that's kind of the earliest stage that he's showing in time. And he's trying to make the point here, look, they're just really, really similar. And if you look at the fish, the top left one, you can see these 4 or 5 lines kind of, that's right near the eye. Those representing what's going to turn into the gills. And so the claim here is that, yeah, they all have this. And so even the human has this. And so the human embryo has this gill system or gill slits. Very powerful evidence. Okay.
I still hear this today. This is still talked about today, even though it's, totally debunked.
So Here is page 449 that I was talking about from Darwin's first edition, 1859. The structure of the embryo is even more important for classification than that of the adult. So he's been making that argument. For the embryo is the animal in its less modified state. So I pointed that out, this phrase, less modified state, a very important concept that Darwin has slipped in. And he's at this point at chapter 13, this is now something he wants to refer to as a given. So he doesn't have to really make the case. There is this thing called a less modified state. And if you agree with that, then you've got evolution already. For the embryo is the animal in its less modified state, and insofar reveals the structure of its progenitor. We may feel assured that they, like two different species, have both descended from the same or nearly similar parents if they pass through a similar embryonic stage. So continuing on page 449, thus community in embryonic structure reveals community of descent. We can clearly see why ancient and extinct forms of life should resemble the embryos of their descendants. Extinct forms should resemble the embryos of their descendants. So this all sets the stage. Darwin is really laying out what Haeckel kind of canonized as ontogeny recapitulates phylogeny. By the way, these ideas do predate Darwin also. It's not like Darwin just totally came up with all this. I mean, before Darwin, there were naturalists, as I mentioned at the outset, in the earlier 19th century, who did look at these similarities between embryos in different species, and they noted them as homologies. They said, yeah, these pharyngeal pouches in the fish and the human, they are homologous, but they're using that term in a classical sense, not in an evolutionary sense. Although some of them may have had evolutionary tendencies, and some not. But Darwin was the one who really, laid out this framework of evolution in a big way, and then he was able to incorporate different ideas, one of them being embryology. So the earlier embryological work he fashioned and fit into his framework very nicely. And here on page 449, it's really coming together for him. So that is that. So let me move along if there's anything else I want to say here. Okay, I said that. But right there,
Go ahead.
Right there in the embryo, you have a highlight reel of evolutionary history. It's so tantalizing. So what was your question, Rebecca?
Oh, it's just, so you're saying that the word homology for back at that time just meant a similar structure. It didn't have the thing baked into it of being from a common ancestor.
Right. But it did have something baked into it, and it was a little more subjective. Naturalists recognized that there were different categories of similarities between different species. Some similarities just kind of looked different. There's probably a better explanation that I can give. But so it wasn't just that all similarities were homologies. There were certain kinds that kind of seemed like they were special, and they called those homologies. And so those Darwin, poured evolution concept into. What a tantalizing concept. So then we have Ernst Haeckel. Here's Ernst Haeckel. Ontogeny recapitulates phylogeny. And he had these sketches, as I mentioned. And this is George Romani's version. There's the pharyngeal arches in the fish embryo that we talked about. So there's a blow up there. Pharyngeal arches in fish embryo will develop into gills. That was known. That's a scientific observable. The pharyngeal arches in the human embryo will develop into head and neck structures. They don't develop into gills, of course, but they are referred to. Darwin had claimed an evolutionary relationship. The human embryo and the adult fish had this relationship of common descent. Here's a later claim that Darwin makes. We may cease. Oh, no, let's just skip that. We already talked about that. I don't want to spend too much time here. The relationship became canonized in Haeckel and later evolutionists. Okay. Darwin approvingly cited Haeckel in his 1872 edition. Yeah, I'm going to some. This became an icon of evolution. I think that's safe to say. This especially the gill slits, but the whole idea of ontogeny recapitulates phylogeny. Okay. So let me show you. We've talked about the 19th century. I know people will say, "Well, that's 19th century," So let's show some more recent contemporary examples. Here's one from Jerry Cohen, 2009, Why Evolution is True. Yet we shouldn't throw out the baby with the bathwater. So he talks about how, there was this problem with the whole ontogeny recapitulates phylogeny issue, the concept. But let's not, drop it all together. Embryos still show a form of recapitulation. Features that arose earlier in evolution often appear earlier in development of today's species. Now, that is not a scientific observation. That is, he's stating it as though it's a scientific fact, because it's some sort of empirical observation. Yes, we got to be careful here. It's evidence, and he's presenting it as evidence, and it's powerful evidence for the theory. But we got to be careful with this. I'll talk more about that in a minute.
But can you explain what you mean when you say it's powerful evidence? Because I think people get confused when we just throw it out there as powerful evidence, but what does that mean?
I mean, I'm going to criticize it in a second and show how it fails. But if you ignore the failure, evolutionists are pouring in other things into this observation. They're ignoring the problems, and they're taking it as, look, if you're sympathetic to evolution, if you think evolution is true already, this really piles on. I guess that's a way to understand what I'm saying here. If you're already, you're already thinking evolution is true, you're going to be this will seem very powerful.
Yes.
Yeah. Here's a quote this from Jerry from his book. Continuing, skipping down. All vertebrates begin development looking like embryonic fish, because we all descended from a fish-like ancestor with a fish-like embryo. We see these gill slits. So he says we've got gill slits in our embryo. Tim Barra, 1990, in his book, he talks about gill slit systems. What do you say? A gill slit stage. That's what he talks about. We pass through a gill slit stage. He uses the word stage. So we've got gill slits. We've got a gill slit stage. Elliot Sober, human fetuses and fish have gill slits. Sober is a philosopher, but he is heavily into evolutionary theory. So he's not ill-informed. He knows what's going on here. And he's claiming that we have these gill slits. Ernst Mayr, yet in certain features, as in the gill pouches, the mammalian embryo does indeed recapitulate the ancestral condition. Ernst Mayr, one of the top, if not the top, evolutionists from the 20th century. Carl Woese was maybe more influential, but anyway, you get the point. This is not a tangential figure here. So he's making this claim in his 2001 book that mammalian embryos have these gill slits. Here is, I mentioned that I happened to run into I wasn't even looking for it specifically. I was just searching for an embryonic page, and I found here's a website presenting evidence for evolution, and they showed Romane's drawings from 1892 as this evidence. So you would think by 2026 we would at least, if this was true, wouldn't we have a better version? We're depending on a very old rendition here in 2026 for evidence for evolution. But there you have it, George Romane's sketches. So embryology is canonized. Okay, so that's in a nutshell. There's a lot more to say about evidences for evolution from embryology, but I'm trying to make the evidences that are most common, most powerful for evolutionists. And so now I want to talk about the problems with that evidence. And there are substantial problems, both with what they're saying and what they're not saying. Regarding what they're saying, those gill slits, that did fall apart. It does fall apart. The human embryo doesn't have gills. It doesn't have slits. It doesn't have gill slits. It doesn't have a gill slit system or a stage. It doesn't have any of this. It does have these pharyngeal pouches and pharyngeal arches and so forth that you saw. Yes, it does have those. But the the draw is so strong. It's so tantalizing for evolutionists when you are coming at it from that perspective. This is like confirmation testing. Your confirmation bias. You're interpreting it through a lens of evolutionary theory, and so for you, it is a homology. And so therefore it is like a pre-gill structure. It will turn into a gill in the fish. In the human, it has now been rejiggered and re-engineered. So it's not going to turn into a fish, but there you have it. It reflects that history. And it is such a powerful, tantalizing concept that they can't let go of it. And so it continues in the literature. You saw it right in contemporary literature. It doesn't work. So the whole gill slit story and the ontogeny recapitulates phylogeny story fell apart and just doesn't work. Mainly because, from several reasons, but the human embryo, the mammalian embryos simply don't have that. That's not what the embryo has. So not a good sign, but if you take away a powerful argument, that doesn't mean it's still not a good evidence. So what else? There are a lot of much more powerful arguments against this embryology serving as evidence for evolution. In fact, what we're going to see is it's evidence against evolution. And I'm going to tell you about three things. First, epigenetics. So when you have an embryo, the first thing that you have is the zygote. It's a single cell, right? That's when the sperm and the egg get together, forms a zygote. That's the new individual, right? You have a zygote. It starts to divide. You get 2 cells, 4, 8, and so forth. It forms, those cells, as they multiply, form a sphere, a hollow sphere. Really interesting. It's called the blastula. And so you got this hollow sphere. It then, one side starts to push in and it forms kind of like a cup. And that's the gastrula stage. Gastrula stage. So you got the blastula and gastrula stages there. And in those stages, all of the cells are identical in the sense that you could swap, theoretically, if you swapped 2 of the cells, it would not harm anything. The individual would still develop. So the cells have not differentiated. They're all just multiplying cells of this young little individual. But after the gastrula stage, that is where you start to have differentiation. And as this thing starts to grow and take different forms, these cells over here, they're going to become, the neural system, and these cells are going to become the skeletal system, and these are going to become, the head and neck and so forth. So you can't swap at that point. If you swapped 2 cells, you'd screw things up. So that differentiation is occurring, and you're losing potency. Embryologists use this term of art, potency. When you have at the blastula and gastrula stages, you have total potency. All cells can become anything. But at later stages, as they differentiate, now these can only be, they can only become this part of the adult or of the fully formed individual. And so they're losing potency. They're becoming differentiated and specified. And so you can't just switch them around. And so this has been known for a long time, right? Embryologists have been doing these experiments and learning about these structures and the potency loss and the differentiation. These are classical concepts in embryology. These have been known for a long time. But when it was discovered, embryologists and evolutionists didn't know how this is happening. Like, how does the differentiation occur? We still don't know it completely, of course, but we do know something about it. And one of the important mechanisms for how these cells differentiate and become neural system or whatever is epigenetics. Epigenetics is, in a nutshell, the turning on and turning off of genes in the genome. So at the blastula stage, you had this, each cell has a genome and they're all the same, but in later stages, after the gastrula stage, after they start to differentiate, those genomes start to become different. The ones that are destined, that are fated to be in the neural system, certain genes are getting turned off. The ones that are destined to be skeletal, certain genes are being turned up, but they're different genes. So the epigenetics are different. The epigenetics controls the genome. It's kind of like the driver of a car. The genome is the car, the epigenetics is the driver. It's turning on and turning off different genes. This epigenetics is a very important thing in biology, in molecular biology, cellular biology, that we've only known about it for about 40, 30 years now. And so now we understand that epigenetics is a super important concept and mechanism in embryology. So before the 1990s, it was like, what's exactly going on here with differentiation? How does it do that? Well, it's epigenetics. So when you read the contemporary literature today, it's epigenetics all over the place in the embryological literature, and as well it should be. And they're devising new and better experiment methods to fish out and parse out what's going on. What are the epigenetics doing? So epigenetics is turning on and off genes by attaching little organic molecules to the DNA or to the histones about which the DNA is wrapped. So you get these little signals, it's like barcodes, little chemical signals being attached and moved around and removed and so forth. So proteins are doing this. It's a fantastically complex system. And so these proteins are doing this because of signals that they're receiving. So there's signals in the cell and coming from outside the cell that are influencing these proteins to go attach a methane group right there at the DNA or at the histone tail. Very, very interesting. Well, the catch to all this is that evolution fails on epigenetics. In fact, evolutionists and evolution denied and refuted and rejected epigenetics for the better part of the 20th century. As I said, it was the 1990s is when we really learned about epigenetics, but there were hints long before that. And it all falls into a concept of that Lamarck, back in the 19th century, before Darwin, advocated. And so evolutionists hate Lamarck, and he's considered to be, his system, his method, his idea for evolution was considered to be teleological. And so that was off limits for evolution. So if you, in the 20th century, if you were a life scientist and you mentioned Lamarck approvingly, or his ideas of like the inheritance of acquired characteristics approvingly, if you wanted to base your research on that, if you wrote a grant or a technical paper, or a journal paper, or gave a lecture or anything like that, you were putting your career at risk. It was scathingly criticized. It was the third rail in the life sciences. You don't touch it. That's the third rail. It's electrified. So evolution and evolutionists scathingly criticized and rejected epigenetics, even in the 1990s and into the 21st century, they continued to do that. They continued to be in denial. I know this from the literature, but I also know this from personal experience in my own personal debates with evolutionists on college campuses against evolution professors. And when I presented in the 2000s, I presented epigenetics as a powerful refute contradiction to evolutionary theory. And I learned that they rejected it and denied it right there in the debate. Not only had they denied it in the 20th century throughout, but right there in the debate, there's no, Hunter doesn't know what he's talking about. You're over exaggerating, you're misinterpreting just some minor findings from 30 years ago. You're exaggerating. And I'm like, I knew that was not the case, but it's that it become your reputation against that guy's reputation. You're in the middle of a debate, and you can't rely on your debate partner to be be up to speed and following the scientific evidence and the scientific findings. That's disappointing, or they're just making refutations.
Yeah, and it actually seems like they're still rejecting it at a level today because they still don't want to admit when changes seem intentional. Like,
right.
And at whatever level the intention is there, they don't want to admit that. They want to keep it as this was random mutation plus natural selection that made this change happen, even when it happens repeatedly in, a variety of situations or organisms.
Epigenetic
seems clear. Yeah.
Epigenetic systems are way too complicated for random mutations and natural selection to create. They're beyond the capture of random mutations. So that's a problem right there. But there's another problem, which is even if they did somehow luckily astronomically evolve, just very unlikely, they wouldn't be selected for, because epigenetic mechanisms are for future conditions. They work in the future, and natural selection needs it to work now, right now. If it doesn't work right now, it will not be preserved. So that defies evolution on that count. So it's never going to evolve in the first place. It's way too complicated, way too many moving parts that you can't put together just by random mutation. And then it's not going to be selected for. But then, as you said, Rebecca, it smacks of teleology. The organisms are responding to needs intelligently, very quickly. So when you have an external environmental shift, an environmental challenge, the majority of the population very quickly and simultaneously changes. So within a generation or two, the majority of individuals in the population make this change. That's not random mutation and natural selection. That's a designed change. So it's not surprising that evolutionists throughout the 20th century scathingly criticized and rejected and fought against this concept. And then it became no longer deniable in the 1990s. At that point, evolutionists they minimized the importance of it. It's okay, there is epigenetics, but it's only like a physiological thing. It's intra-lifetime. It's not inter-lifetime. It's not transgenerational. It's not passed on to the next generation. It's just within your lifetime. Well, of course, that's false. That's another one of the marvels of epigenetics, is that it is passed on into the germline. So environmental challenges that you are presented with can be passed on through your germline, which is astonishing. Again, something that would never evolve by random mutations. It's just an incredible technology that, your egg or your sperm is going to be influenced by this, so that your progeny is then going to pick up on these messages that of the environmental shift or challenges that you faced. This way beyond the evolutionary capability. So evolutionists said, "Oh, it's only within a lifetime." Well, no, actually, it is transgenerational. Well, if it's transgenerational, it's not robust. It can only be passed on for a couple of generations. No, actually, it is robust. So everything they've said, they've been pushing back on it, and they continue to be rejected or contradicted by science. So here you have in embryo. So consider that whole. I had to kind of give a quick review of Lamarck and the inheritance of acquired characteristics and epigenetics. with that in mind, now understand it's crucial, it's fundamental. Epigenetics is foundational in embryology. Embryonic development is absolute. The differentiation process of those cells is all using epigenetic mechanisms. Epigenetic mechanisms are driving differentiation. I mean, I'm not saying they're the only mechanism, but it's a very important mechanism. If you look in the literature, epigenetics is all over the place. So if you're claiming, now let's ask the question, how does embryology reflect on evolution? Oh, embryology incorporates epigenetics. Epigenetics is crucial. I don't think I think I should distance myself from embryology. I don't want to be saying embryology and evolution in the same sentence here, because we are contradicting evolution, because we have to have epigenetics. Epigenetics is now dominant in the embryology literature, and yet evolution has been rejecting and refuting and denying epigenetics. So you got a real problem here. If you're going to claim embryology as a key evidence for evolution, that's a real problem. So that's, the first of these problems. Another one is the germline itself. So when an embryo is doing its thing, it's building the body of the new individual, it's also doing something else. There's something else going on the side. It's building the germline. So that new individual is going to have the procreation capability, the capacity to have children. And so it has to have its own germline, right? If you're a female, that's going to be eggs. If you're a male, that's going to be sperm. But you have to have the capacity, you have to have eggs or have the capacity to make sperm. And so in the embryonic development, that's going on too, the building of this germline. And so it's a fantastically complex thing. So consider this diagram. You have the egg and the sperm. They get together, make the zygote, make the new. Then you have embryonic development, making the new individual. But then you have egg and sperm coming back and to complete that diagram from the new individual. Whether it's a male or a female, it's going to have egg or sperm to contribute. It's going to come back and make in the next generation. So it's kind of a cycle. Well, that whole process of making the new germline during the embryonic development, it's wildly complicated, incredibly complicated, of course, but it's also not, it doesn't fit the evolutionary tree. So turns out there's different modes. There's two primary modes of creating the germline. And they're not clustered in the evolutionary tree. It's not like, the mammals are one and the fish is a different one or something like that. They're sprinkled around. And so if you're an evolutionist, you have to say that, well, evolution created this fantastically complex capacity to build the germline, but then it changed its mind and, did a different method and then switched back to a different method. And so you look at the evolutionary tree, it's all sprinkled around. There's no clustering. And so it's not homologous. You don't have the creation of these. And by the way, the germline a key phase in this germline process is the creation of primordial germ cells, PGCs, primordial germ cells. They're a fundamental part of creating the germline. So this PGC creation is where you have, different modes, and you have to say that, it just did one mode and then it switched back to the other mode and it switched to this mode. And it doesn't make any sense on evolution. So the homology doesn't work. It's not consistent with the evolutionary tree. And so that's a problem. If you're going to claim that, the embryonic evidence is powerful evidence for evolution. Well, wait a minute. What about this germline creation that's so important in embryology? That's not fitting the evolutionary tree. Well, that's just PGCs and germlines. That's a special case. well, it doesn't matter if it's a special case. It's still a problem for evolution. But it's not the only case. And so that brings us to the last problem, that last major problem that I'll talk about, and that is just non-homologous development in general. So when you have the embryo developing, it creates the brain and the neural system and the eyes and everything, right? There are different ways that you can create these things. So there's different modes. For the classic example is the eye in two species of frog that were discovered early in the 20th century, about more than 100 years ago. These two different species of frogs, the eyes are developed in different modes. There's different ways to create the eye. And evolutionists thought, well, all frogs have a relatively recent common ancestor, the frog common ancestor, and that frog had eyes. So you're going to have the same embryonic development mode, the same embryonic development pathway. The same mechanisms are going to be used. If you look at all frogs, they're going to make their eyes in the same way in the embryo. That's not the case. Again, you see different modes, and it's sprinkled around the evolutionary tree. Why would that happen? I'll talk more about that in a second, because I'll show a graphic to kind of pound this home. But it's a very important finding in biology in the 20th century that demolishes evolution. You can't have evolution with this. It just doesn't work. Without it, you would have to resort to ridiculous explanations. So let me go through a few slides and show you what I'm talking about.
Well, and that is what they've done. They have resorted to ridiculous explanations because they seem to have no problem with these trees that have all these different, like you said, with the frog eyes, okay, you'd think that they'd all develop the same way. They don't.
Yeah.
And it's not just frog eyes. It's like there's so many things, like every tree has those kind of problems.
Right. But at the embryonic level, it is also not just frog eyes. It's a ton of examples. That's kind of the poster child example. It is the rule rather than exception. So at first, evolutionists would have said, well, that's frog eyes, but that's a rare example. No, it isn't. It's the rule. So you have this non-homologous development pathways throughout biology. It makes no sense on evolutionary theory. Here's the gill problem, and here's an illustration of the human embryo and those pharyngeal pouches that you see. They're not gills. Here is an artist rendering of the gills in fishes and how the gill system works. It's really complicated and fantastically efficient. So I won't belabor the gill problem here. This is an interesting book by Paul Ehrlich and Richard Holm out of Stanford, 1963, talking a little bit about development. And he's got some interesting quotes here. Let me read a little bit of this. Of all the phenomena of morphogenesis, none has received more attention from evolutionists than so-called recapitulation. So that's Haeckel's idea, Darwin's and Haeckel's idea, recapitulation. Ontogeny recapitulates phylogeny. It was soon observed by embryologists that early developmental stages of vertebrates resembled one another, at least superficially, to a much greater degree than did the adults. Yeah. Okay, so we talked about that. This has been interpreted by some workers to mean that in the course of development, each organism goes through a condensed version of its phylogenetic history. That man, for instance, goes through a one-celled stage, the zygote, fish stage with gill pouches, and a mammalian stage. This generalization was originally called the biogenetic law by Haeckel and is often stated as ontogeny recapitulates phylogeny. This crude interpretation of embryological sequences will not stand close examination, however. Its shortcomings have been almost universally pointed out by modern authors. but the idea still has a prominent place in biological mythology. Now, this is an evolutionist writing here, so it's not it's a sympathetic author, clearly stating the problems here. The resemblance of early vertebrate embryos is readily explained without resort to mysterious forces, et cetera, et cetera. Okay, then there's some other quotes, some other papers talking about this. Okay, let me read the first quote. The so-called basic law of biogenetics is wrong. Not buts or ifs can mitigate this fact. It is not even a tiny bit correct, or correct in a different form, making it valid in a certain percentage. It is totally wrong. So good refutations here. Okay, that's that. Now the epigenetics, I talked about this blastula and gastrula. So here's an illustration. At the left, you have the zygote, the single cell. It starts to multiply. It forms this sphere called the blastula, and that one side kind of pushes in and creates this cup-like structure called the gastrula. And then after that, differentiation starts. This is just the very beginning. I mean, there's obviously incredibly complex. We could spend, it's a whole undergraduate class, a 3-credit class, just to even scratch the surface of embryology. But differentiation begins after that. Epigenetics plays a crucial role, but yet evolution denied it. So we talked about that. Okay, I have a lot of papers here, and I'm not going to read from these. These are papers, contemporary papers in embryology, discussing what epigenetics is doing, and it is a lot. In mammals, the fusion of sperm and oocyte gives rise to a totipotent zygote, which undergoes a series of cleavage divisions and differentiation events. During this process, the embryo transitions from totipotency to pluripotency, accompanied by extensive epigenetic reprogramming. So just one example of the importance of epigenetics. And I'm just going to skip through a dozen or so of these that I'm storing here in my file for my reference. It would be too long to read through them, but it's just to give you an idea of the importance of epigenetics. You can see these papers. It's crucial, the crucial role. It goes on and on. What evolutionists have rejected and denied is crucial and fundamental to embryonic development. You simply cannot blithely claim, oh yes, embryonics and embryology is powerful evidence for evolution. That it's just a ludicrous claim. You're just really turning you're either ignorant or you're just not being honest with the evidence. And then I talked about the germ lines and the PGCs. Embryonic development also creates the germ line. So I talked about that in this. You get the sperm and the egg, the zygote, embryonic development, but then you have these PGCs, primordial germ cells, that's either going to create a sperm capacity to have sperm or the eggs. The germline is core to life. It is so crucial to life, and yet it's convergent. Well, let me skip forward to an illustration and illustrate this. I'm going to skip through these slides here too. I have an illustration here. There's an illustration. I'll zoom in on this. So these are insect groups on the right-hand side, and the green boxes and the red boxes have different modes of the creating the germline. So if you're an evolutionist, you have to say that, well, way back at the common ancestor, it was the induction mode that had evolved. But then later, the inheritance mode evolved. But then within that group, evolution reverted back to the induction mode in these three different groups. So again, these two different modes, not only is it beyond random mutations and natural selection to create them, but they had to be sprinkled around. They're not aligned with the evolutionary tree. So that doesn't make any sense. So then you have these just so stories to try and explain it. Okay, but it gets worse because it isn't just insects. It's a lot more. So the induction mode is found in crickets and mice and salamanders. The inheritance mode is found in flies, wasps, nematodes, and frogs. Again, sprinkled around in the evolutionary tree, these two different modes, induction and inheritance. So it's lineage specific. You find these modes, the germline modes is lineage specific. And that's an antithesis to evolution. The whole concept of lineage specific is antithetical to evolutionary theory. Evolutionary theory works off of common descent. That's the whole idea. You have these clades, and within the clade you have the commonality. You don't have differences within a clade, and that's what we see here. It's lineage specific. But it gets worse because even within each mode, the designs are lineage specific. So for the inheritance, the induction, they have different genetic genes at work, different ways of implementing that mode. And so again, at a lower level, you have another misalignment with the evolutionary tree. Yeah, I'll skip that one. Evolution. There's some teleological language as usual. Okay, so germline summary. Germlines are fundamental in the development of animals. Germlines arise in two different modes. They are lineage specific with details within each lineage specific clades. But it gets worse, non-homologous development pathways. So when you have two species, like two species of frog, this is kind of a notional evolutionary tree. At the top, you have the extant species, the species we observe today. So think of the red box as containing two different species of frog, and they're going to be very similar. That's why they're the same. They're in the same genus or family or order. So they have a lot of similarities, right? And they're going to have a relatively recent common ancestor, yet fundamentally different embryonic development. So you have this relatively recent common ancestor. That common ancestor looks a lot like the extant species, but yet when the eye is formed or different structures are formed, it's differently in the embryonic pathway. They get formed through different pathways. So in that little piece of time there, since the common ancestor, evolution would have had to have preserved the adult structure. You have the eye or you have whatever it is, but it changed the way that it was created. That doesn't make any sense. There would be no reason to do that. And also it would be way beyond the means of random mutations. You're disabling a embryonic development pathway and replacing it with another one. So here's two frog species and two notional frog species. So think of the time. You see the time arrow. Time goes up, and at the bottom of the screen is way back billions of years ago, and evolutionary history plods along over billions and billions of years, and you get to the point where you have this common ancestor. You have this frog, and you have a split here. And some relatively short time later, you have what we observe is these two different frog species. So what you're saying is that, well, let me go to the next slide here. So there's a long, long evolutionary history where these two frog species share the same history. They share the same history, and only a short little time where they are evolving independently. Adult structures are conserved. During that short little time, the adult structures are conserved, but they develop differently.
Wow.
So yeah, wow, exactly. So evolution would have radically changed the embryonic development but not the final result. It would have gone in and done this differently. The science contradicts the theory. So I have quotes from several evolutionists here talking about this. And here's one example, this canonical or poster child example of these two frogs. So I'll talk a little bit about this one. Sir Gavin de Beer was a British embryologist. Note the Sir, like this guy's a serious player. He was a Sir, and did a lot of good embryology work for the majority of the 20th century. There are many. So what I have here is a synopsis of some of his discussion of these two frogs and the development of their eyes in his 1970 pamphlet, Homology and Unsolved Problem. And remember, this guy's an evolutionist, true evolutionist. He doesn't doubt it for a second. There are many examples of closely related species with unexpected embryonic differences. For example, the frog species Rana fusca of Southeast Asia develops its eye lens from the epidermic on the optic cup. It is the optic cup that induces the epidermis to differentiate into the lens, which ends up perfectly fitting. It makes sense that the lens perfectly fits into the optic cup since it is developing from the epidermis. But in the closely related frog Rana esculenta, the optic cup can be cut out from the embryo and the lens develops all the same. It cannot be doubted that the lens of these two species of frog are homologous, yet they differ completely in the mechanism by which determination and differentiation are brought about. In other words, by the embryonic mechanisms by how they were created. This is no isolated example. All this shows that homologous structures can owe their origin and stimulus to differentiate to different organizer induction processes, blah, blah, blah. So he's an evolutionist. He still thinks this is evolved, but he's admitting that, somehow they were created separately. Here's a graphic that he has in there. What mechanism can it be that results in the production of homologous organs, the same patterns, in spite of their not being controlled by the same genes? I asked this question in 1938, and it's not been answered yet. This is 1971. So this has been known for most of the 20th century. So he wrote this little pamphlet in 1971. A few months later, he passed on. And then he learned about embryonic development. But at this stage, he was still casting it in terms of evolution. Here is a book or a chapter by Hall, and I won't read all these quotes, but he's saying the same thing. He's saying this is just it's prevalent. It's not just isolated examples.
Okay, I want to stop and make sure people understand, because you did cut out for me on the quote that you were reading. It may not be cut out on the main thing, the main recording. But so what you saw in that quote is that the resulting structure is the same, but the embryological development is different.
Yeah.
So that is a mess. It's a contradiction for evolution. It's a problem for evolution, because you have to get the same result through a different pathway. And so it's not following the evolutionary tree of life pattern.
And it's also not following the evolutionary logic. This is not expected by evolutionists at all. And for good reason. Just think about it. You have a species some time ago, and then you were saying, I now have its descendant, which is very similar. But during that period of time, I swapped out the embryonic development pathway and swapped in a different one. So the way I'm going to create the brain or the eyes or the jaw or something, I'm going to do it a different way. That would create a mess. I mean, just, you can't just take the one down and start to evolve the other. You have to continuously have that in place. Right? You can't turn one off and then evolve the other one. It takes some time to evolve another one. So why would natural selection or random mutations generate a different method of doing it when you already have one that's working?
Yes. And so all the while they're trying to convince us that similarities in appearance, superficial similarities in appearance in embryos are evidence of our evolutionary history. But even at the tip of their tree, they have these major contradictions in the embryo development.
Exactly. Yeah.
Wow.
It's a huge, huge refutation of evolutionary theory. And here, this chapter, Hall is explaining. This guy's an evolutionist, but he's admitting this just doesn't really make sense. That congeneric species, what's congeneric? That's different species in the same genus. So they're pretty similar species. Congeneric species exist in which one species shows induction and one self-differentiation, two fundamentally different developmental mechanisms. Surely lenses in eyes of congeneric species are homologous. So the whole idea of they got to be homologous, that means they came from a common ancestor, but the way that they're created is not homologous. So we call this non-homologous development pathways. And it's like looking under the hood and finding out that, oh, it wasn't what I expected. So when you first look at the biology back in the 19th century with Darwin, it's like, oh yeah, these frogs, they're just the same, and these different species are just the same. And now we know that they're not the same, but at a deeper, more complex level. Here's another paper by Gunter Wagner. The more examples of homologous organs with mechanistically different modes of development become known, we can safely assume that some level of variation in the developmental mechanisms of homologous characters is the rule rather than the exception. Again, this guy's an evolutionist.
Yeah.
Continuity of morphological characters is not subscribed by continuity of genetic information. In other words, if you have similar visible structures in different species, that doesn't mean you have similar genetic information driving their embryology. This seems to be a pretty depressing situation. If one thinks that homology should somehow be related to the evolution of development and motivates the notion that homology may be an illusion, this is motivating the idea that homology may be an illusion. The theory is just falling apart. But these guys will never come to that conclusion. Here's another example: tomatoes, and there's a plant called bittersweet. If you've ever heard of that, they're similar. Bittersweet and tomato are similar plants. The pollen-producing anther organs of both of these closely related species are both arranged as a cone. These cone structures appear only in a single clade within the genus, so it's not that common. They would have traditionally been thought to derive from a common ancestor, and so have similar embryonic development pathways. But whereas small hairs known as trichomes are required for the cone formation in the tomato, in bittersweet, the trichomes surprisingly prevent the cone formation. These two species produce the same macroscopic structure through two mutually exclusive developmental routes. It's just another example that I pulled out to show you. I could do this all day. It is the rule rather than the exception. Here is a couple of quotes from Rudolf Raff, another leading researcher in embryology from 1999. The process of early development from the egg to the phylotypic stage, that's an intermediate stage in the embryological development. So it's a stage where they kind of tend to look, different species look alike, different vertebrates tend to look alike. That's the phylotypic stage. The process of early development from the egg to the phylotypic stage should be at least as conserved as the pattern of the phylotypic stage. What he's saying is, if the phylotypic stage they look alike, then the embryonic pathway to get there better be conserved. They better look alike also. He's just saying the embryonic development better be conserved if what you're getting to is conserved. And of course, it's not. Now, this is a paper, I'm quoting, this is a paper by David Swift. He's a British guy. And this is an example of a guy who's willing to go there. And he's willing to say the obvious. And so it's actually a very good paper. As I say, all the others that I've read from so far are from evolutionists. And this guy, I don't know where he stands on the spectrum or anything. I'm sure he was probably an evolutionist, and now he seems like he's questioning it. But he's doing it is because of this evidence. And so it's a very good paper. So I want to read a couple of quotes from this paper here. However, despite their embryonic morphological similarities, and contrary to evolutionary expectations, the striking fact is that the phylotypic stages of different groups of vertebrates arise in remarkably diverse ways. So this he's talking about the early stages, the early parts of the embryonic development. They are remarkably diverse. Even with key tissues such as the germ layers deriving from completely different early embryonic structures. These observations clearly refute the presumed evolutionary homology of the vertebrate phylotypic stage, and hence undermine the inference that common ancestry based on that supposed homology. So he's pointing out the obvious here. You've got these different embryonic pathways leading to these hackle embryos, and they're just different. The embryonic pathways are so different that this is undermining the inference of common ancestry. Especially significant from an evolutionary perspective is that the diversity extends from the earliest stages, zygote and blastula and gastrula. He's going to talk about that. This apparent homology is refuted by more detailed embryological evidence. Despite their similarities, the phylotypic stages are formed embryonically in profoundly different ways. The straightforward conclusion to draw from this radical diversity of their early embryonic development is that it shows the vertebrates have not evolved from a common vertebrate ancestor. So he's willing to go there, whereas the other evolutionists have resisted taking that step. It's just a straightforward conclusion. It's like modus tollens. If evolution is true, then you would have these homologous development pathways. If the development pathways are not homologous, then evolution is not true. Then the common descent pattern is failing. He wouldn't say evolution is false, probably. He would just say it's not a common ancestor. You'd have to have, these must have evolved separately. I'm guessing. I'm not sure what his overall position is.
Well, it's amazing. I love it when, even evolution believing scientists can be very honest about the evidence. So that's one good example. But even when it doesn't register as a contradiction for them, I try to tell people, like, everything needed to falsify evolution has already been published. Like, if we're talking about the scientific theory of evolution, everything to show that it is false is already published in the secular literature. You don't even need creationists to go after things and publish papers, because the evolutionists have already done it themselves. You may have to read between the lines, because they're always going to, because they believe the paradigm, they're always going to say, but we'll have ways of working this out or whatever. But
they will always cast it in an evolutionary framework.
Yes. And so it's really good, though, when you find papers like this where somebody is being honest about the evidence, even if they're still sticking with the paradigm.
Yeah. Here's another paper that talks about this fact that early animal embryos are not highly conserved. That's a nice pithy way of saying what I've been talking about the last few minutes. Early animal embryos, that is embryos in their early stages, are not highly conserved. I think that's what it means. Okay,
Yeah.
so I want to bring everybody back to the beginning of this podcast. What did I say at the beginning? I said Darwin had a few key observations, but there was one that was really the most important. And the one I want if you remember nothing else, remember this one. And what was that one?
Yeah, it's the resemblances are the important thing. But before we go there, can we just talk about one thing that we haven't talked about so far with the scientific stuff? Because I know you're about to go into talking about why this still goes on, even though it's been falsified scientifically. But that we didn't talk about the new Evo Devo with the genetics and looking at the genetics. And so, this whole thing about, we see the same genes. Okay, like for example, the PAX6 gene, it helps our eyes develop. It also, that same gene PAX6 is also in mice and in chickens and maybe and even way farther back than that. And so the idea now is that, hey, we can even look and see the development of genes, and we all use these same genes, and they can take a PAX6 gene out of one animal, put it in another animal, and the animal still develop in normal eyes. And so they use this as evidence for, hey, see, we developed this early toolkit of genes and they have continued throughout history. And this is evidence of evolution and things like that. So I just wanted to bring that up because I know that's a big deal. And it is cited as, this powerful evidence for evolution. And I want to just give you a chance to respond to that, Dr. Hunter.
Yeah. Okay. Thanks, Rebecca. So Evo Devo came on the scene, a couple decades ago or so, and had high promise, high hopes that Evo Devo was going to resolve these emerging issues that people are starting to see with embryology. And that has failed. So there was the high hopes have been dashed. Evo Devo never produced never lived up to its expectations. So that's why it's not really worth talking a lot about it, because it's just there's not a lot there to talk about. If anything, it would be, well, okay, epigenetics. We now know, yes, there are really important genetic maneuvers going on with epigenetics. That does not help. That hurts evolution, as I talked about a few minutes ago. Now, you mentioned PAX6. It's a master control gene. So it's a gene that can turn on many other genes and turn on pathways, turn on entire metabolic pathways, or embryonic pathways. Sorry, it can turn on entire embryonic pathways. So master control gene, very important in embryological development. And like you said, you could take the PAX6 gene from one species, take it out and put it into another species, and it works just as well, even though the PAX6 in that other species is slightly different. It has a different amino acid sequence. It's not that different, but it's different. But you can swap in the gene in PAX6 from some other species, and it works just fine. And that seems like, they must share a common ancestor, right? Well, no, not necessarily. But one problem with that logic is PAX6 is so widespread, and it is used to build different types of vision systems. If you look at the vision systems in biology, there's many different types. The fly has a very different, it has a compound eye, very different kind of vision system than, say, humans. So PAX6 is so widespread that, remember, the evolutionary tree and the rule here, it has to come from the common ancestor. So you've got PAX6 in a whole bunch of species. You go to the common ancestor in the evolutionary tree, of all those species, and PAC6 had to be there. Well, at that common ancestor, there was no vision. So PAC6 had to be present in an organism where its job is going to be, among other jobs, building vision systems, but there's no vision system in that common ancestor. Well, that doesn't make a lot of sense. How do you explain that? Oh, we call that pre-adaption. You have things evolving before they're needed, and they might be used for they have other uses, and then we find that it gets used for vision when the time comes. Oh, so it's serendipity. No, creationist, it's just good, solid science. You see my point? The story is it's just a tautology. Whatever we find, it evolved. It doesn't make sense under if you're an evolutionist, certainly never predicted this. This is not a prediction of evolution. No one ever said, yeah, we expect to find these genes before they're needed, and they're going to fulfill roles, crucial roles at the time. It looks a lot more like something that was designed instead of something that has this common descent pattern. So, yeah, I have a whole bunch of that was another section I was considering talking about was the master control genes and the pre-adaptation concept and how that works. But it's just these other areas I find to be much more powerful problems with the theory. And when evolutionists argue that that's some sort of a powerful evidence for PAC6 or things like that are a powerful evidence for evolution or for an embryological evidence for evolution, there's really no there. So
yeah. And if anything, it has actually made the situation for evolution way more complicated, because you need a lot more than PAC6 to develop eyes. You need many, many genes involved in developing eyes. So it would be great for evolution if you only needed one gene, right? It would make things a lot simpler. But because you need hundreds or even thousands of genes for developing a certain feature, then you've got to get these genes to coordinate. I mean, mutations to coordinate across genes, across chromosomes. You've got all this mixing and matching. And because the genes do more than one thing, then you've got every change you make is going to affect all of those. So it's just like mind blowing the level of sophistication that we're attributing to random mutations, right?
Yeah, exactly. So, yeah, thanks for that reminder and discussion, Rebecca. Mm-hmm. So we talked about at the very beginning of this podcast, probably the most important observation that Darwin incorporated or launched his thinking on was this resemblances. You see this over and over again in the literature on up to today. These resemblances between the embryos of different species. That drove a lot of this thinking. And Darwin said, in different species, if different species pass through the same or similar embryonic stages, so they resemble each other, we may feel assured that they have both descended from the same or nearly similar parents. So that was crucial for Darwin. Embryonic resemblances were so important, but the resemblances fail when we look under the hood. So that's the problems. The problems with this whole notion that the embryonic evidence and embryology is just this key evidence for evolution. It isn't. If you're going to be honest to the science and reflect to the preponderance of evidence from the science, you're going to say that's a challenge. That's a problem for evolution. It contradicts evolutionary theory. The empirical evidence contradicts evolutionary theory. And when you evaluate a theory, then you're going to incorporate all the evidence you can find, all the predictions and all the evidence, and see what you can see. So no one piece of evidence is going to determine a theory to be true or false. You can collect it all. This embryology, major problem for evolutionary theory. But evolutionists don't skip a beat. And no one has said that, except that one paper by David Swift, where he actually was starting to suggest that. But, and certainly creationists and IDs are, talk about this. But the core of evolutionists, there's no backing off, there's no backtracking off of the embryological evidence. So there's a persistence of the evidence. We saw the powerful evidence, the powerful presentation of the evidence. We saw the powerful failure of the evidence. Now we see the persistence. So what's going on there? Okay, so I'm going to talk about 4 different things, but the basic idea is there are theological underpinnings that's driving this. The first one is the ordinary view. When Darwin wrote about this in chapter 13 and chapter 14 in his 6th edition, he talked about the ordinary view and how the evidence that he's presenting fails on the ordinary view. And Elliot Sober has referenced these passages, this chapter, as an example of how Darwin's reasoning went. And so Sober has always argued that you shouldn't be using metaphysics or religious arguments in science. And he sees Darwin's arguments about the embryology in chapter 13 as scientific. But what's this ordinary view that Darwin is talking about? Darwin has this, the ordinary view fails. Well, the ordinary view is Darwin's way of referring to creationism, or what he would call independent creation. It's a code word. And I have a 2014 paper in the History of Philosophy of Science journal where I go through this and I do a word study on ordinary. How did Darwin use the word ordinary? And it's very obvious because he spells it out in earlier chapters where he introduces this term, the ordinary view, and he defines it over and over again as independent creation. He's calling that the ordinary view. And by the time he gets to chapter 13, he drops the definitions and the explanations. He just uses it. It's just a new term that he's using, the ordinary view. He's arguing that this evidence doesn't make any sense on divine creation or independent creation. And that introduces a whole raft of questions about, how do you define divine creation? Well, that gets into doctrines, theological doctrines, and what you're claiming about what God would and wouldn't do. And there are about a dozen doctrines that are influencing people's thinking about origins and evolution. And Darwin is influenced by those, and he's bringing those to bear. And he brings those out in chapter 13 as well as his other chapters. So this is really a theological chapter. It's a theological book. He's talking about how the science is interpreted from these theological views. And I'm going to show you some examples here. But I wanted to start with this idea that it's the ordinary view, because that is so often lost on people. And if you were to read Darwin's book, if you're not like a historian or a philosopher who's like deep into this stuff, you would never get this. You wouldn't track that, because he just brings it up every once in a while and then you forget about it. And so Elliot Sober, he's a philosopher. He missed it, and it's right there. So it's a exactly what Sober said you ought not to be doing. Darwin is making theological arguments. They are entailed. They are premises in his argument for evolution, the ordinary view. So I'm going to talk about 2 specific theological doctrines that come up, because when you read through chapter 13, you'll see that there are 2 in particular that come up over and over again that Darwin is using. Although they're not easy to see. Again, you have to be a philosopher or a historian to really kind of see this. But the 2 are the principle of plenitude and theological utilitarianism. And the principle of plenitude says God would make the fullest world unless there's a sufficient reason not to. So the only time you're going to see a gap or something missing from the world is if there's a sufficient reason. God has to have a reason. And you might say, well, who are they to make rules for God? Well, this is all based on the goodness of God doctrine. And the Bible says God is good. God is infinitely good. And so if God is good, then as the reasoning goes, he would make the fullest world. He wouldn't withhold. And so anytime you see that something is withheld, then there has to be a reason for that. And so Darwin spends a fair amount of time and spills a fair amount of ink in chapter 13, arguing that there's no sufficient reason for the patterns that we see, these embryonic resemblances. He's arguing over and over again, there's no reason for that. There's no reason. And I mentioned that when we started the podcast, that he had these arguments that there's no reason. Well, that's the principle of sufficient reason. If you can show that there's no reason for something, then you can then conclude that God didn't do it. It must have come about by natural law. So that's going to be one of the branches of argument in chapter 13, is there's no reason, so therefore plenitude, the principle of plenitude is violated. If God were to have created it, it would have been full. It's not full. There's no reason for it. Therefore, it must have evolved. Darwin never spells it out like that. And I don't think he's even probably thinking it through fully. It's just kind of burned into the way we think. And we've been brought up in this world where these arguments are made all the time. So we just kind of naturally make it. Evolutionists today make these arguments all the time. I'm sure they're not even aware of these concepts or doctrines at all. But it's totally, their arguments are totally contingent on these arguments. The argument falls apart without it. There's no science here. So Darwin makes these arguments that there's no reason for these things. The other one I mentioned was theological utilitarianism. If there's no reason for it, and if you have this similarity of design that's not needed for functional purposes, then it's not optimized. If it was optimized, it would be tailored specifically for each species. The term of art here that was commonly used is the conditions in which they are found. So species, the designs of species are not, the question is, are they perfectly adapted for the conditions in which they are found? This was a big debate in France, and Georges Cuvier, one of the greatest minds in the history of science, argued that all the designs of species are perfectly adapted for the conditions in which they are found. So if you have a bird with a goose with webbed feet, that means it's in the water or near the water. And Darwin said, well, actually, we have the upland goose, and it has webbed feet, and it's not near the water, because that's why it's upland goose, right? Not near the water. Yet it has webbed feet. So that's violating Cuvier's claim. They're not optimized. They're not perfectly adapted for the conditions in which they are found. Uh-oh. Well, Geoffroy, as on the other side of the debate, said, no, they're not optimized. There are forms. They fall into categories. There are categories of designs. And so you might have a webbed foot, but that's because it falls into a category. It's just very Platonic. It falls into this category. And maybe it's near the water, maybe it's not. So Darwin says, well, it's because of common descent. It has its webbed feet because it's common descent, but the inefficiencies of evolution are such that it isn't near water now, but that's not a big deal. It can still survive. So his argument is contingent on the claim that Cuvier was correct, that if God created it, it would be perfectly adapted. And so God didn't create it, so therefore evolution is true. He uses this argument over and over again. So you see these two different arguments in chapter 13 also. the plenitude and the theological utilitarianism, where you have these designs, these common designs that are for no reason, therefore they're not optimal. Therefore, God wouldn't have created that. I'll show you some examples here. And then I show you some examples from contemporary evolutionists also making these sorts of arguments. Let's have a look at the ordinary view. Here's the passage where Darwin argues that the embryonic features that we're seeing don't make sense on the ordinary view. But it is by no means obvious on the ordinary view why the structure of the embryo should be more important for this purpose than that of the adult. He's talking about classification. So he's making an argument that this makes no sense on the ordinary view. And most readers would read through that and think, oh yeah, that's a scientific statement. No, it isn't. What is this ordinary view and why is it falsified? It is independent creation. That's what the ordinary view is. And it is falsified by theological arguments, mainly plenitude and theological utilitarianism. The principle of plenitude and principle of sufficient reason goes like this. Embryonic structures at the early stage are often highly identical, and they look nothing like the adult form. Yet there are cases where the embryonic form is similar to the adult form. There are certain species, interesting species, where if you look at the embryos, they look like miniature adults. So they don't go through these stages that we're used to talking about with most vertebrates. So there's some of these oddball species that have this thing where their embryo is like a miniature adult. Darwin latches onto that fact to conclude that could be the way for all species. Any species could conceivably be designed, could be grown that way. The embryonic pathways for any species could be such that it would look like a miniature adult. You don't have to have the different stages that we talk about for embryonic development, because he's got a couple of examples of species that violate that. So therefore, there's no reason for these species to have these constraints, for them to look alike. There's no reason why for the embryonic resemblances. There's no reason for those resemblances. There's no functional reason. So he's working on the PSR, principle of sufficient reason. So the design is unnecessarily constrained. That's a violation of plenitude. Again, he wouldn't use that term. He's just thinking the way they think. But this is all theologically informed. None of this is science. This is all theology. Here's an example. In embryonic crustaceans and in many other animals and in flowers, that organs which when mature become extremely different are at an early stage of growth exactly alike. So again, he's restating this thing. They're exactly alike at this embryonic stage where they're very different in the adult form. How inexplicable are these facts on the ordinary view of creation? Now that obviously is a theological statement. I think everyone would agree that's a theological statement. And I could have started out with this slide, and I could have just showed you this. And I'd make the argument, look, chapter 13, he's incorporating theology into his arguments, his embryological arguments. And that would have been a good way for me to make my point. But I talked to you for the past few minutes about the principle of plenitude and sufficient reason and theological utilitarianism, just so you have a little bit more background. If I just threw this out there, you might kind of wonder, well, why? What's he talking about? So now you have at least a little bit of a hint as to what's going on behind the scenes. He has made these arguments, but there's no reason for these similarities. You've got these identical, exactly alike embryonic stages in these different species. That is inexplicable on the ordinary view of creation. He's making that leap. How does he make that leap? Well, it's what's implicit, what's unstated is the principle of plenitude and theological utilitarianism. Those two different, by the way, those two different theologies often overlap. When you have a unnecessarily constrained design, there's no reason for it. That violates plenitude, but it also violates optimality. It's like, how could it be optimal if I'm unnecessarily constraining this design? You're not picking the best design. Cuvier's idea is wrong. It's not perfectly adapted for the conditions which they are found. So oftentimes the plenitude argument and the utilitarian argument do overlap. They are both kind of the itch is scratched for both of these in a lot of times, and this is a case where I think it's more of a plenitude here, but you could also say it's theological utilitarianism. Theological utilitarianism. Embryonic structures exhibit disutility. Darwin, one of Darwin's favorite words. Because, why? They're not tailored for the conditions of existence, or they're not tailored for the conditions in which they are found. This is an unnecessarily constrained design, so it is not perfectly adapted. So this fits both plenitude and utilitarianism. Here is another quote, skipping down page 439 here, still in chapter 13. It has already been casually remarked that certain organs in the individual, which when mature become widely different and serve for very different purposes, are in the embryo exactly alike. The embryos also of distinct animals within the same class are often strikingly similar. Okay, so he's making this point, and then skipping down. He's. I'm just going to skip to the bottom. He's making this argument, and then he concludes. They have no direct relation to their conditions of existence. So he's pounding home. He's making this argument. There's no reason for this, because there's no relation to their conditions of existence. No sufficient reason. The resemblances are not related to the conditions of existence. And he goes on. He's going to make this point here about the gill slits. He called what evolutionists often refer to them, these gill slits. The more technical term he's using here is the branchial slits. We cannot, for instance, suppose that the peculiar loop-like course of the arteries near the branchial slits are related to similar conditions in these different species. So again, he's making this point. They're not related. They're similar, but they don't have to be similar. And he's continuing with these arguments. I'm going to read through this one. He does mention Cuvier here. That's fitting, because Cuvier was the champion of that view. So it's not surprising that that comes up. And he's going to continue making these arguments. Uh-oh. The slide.
Is it frozen?
It's I'm not sure. I wouldn't say it's frozen because I can go backwards, but I can't go forward.
Yeah, it looks like that's the last slide.
I think it maxed out at 200.
Okay.
So I can talk to the rest.
Okay.
I mean, I could create another file, but I'll just talk to the rest. Let me open up the file on my machine so I can look at it. I'm almost done. So
while you're going through that, I mean, there's a lot of good comments, so I know we've been going for a long time, but
I think the fact that I'm almost through, I should probably just finish here.
Okay.
Basically, I'm just going to, because I'm basically finishing up with Darwin. That was 200. Yeah, he goes on to make several more statements about this doesn't make sense on, the ordinary view or on independent creation. And so I'll skip through his statements and then go to the contemporary literature. I've got, passages from Douglas Futumia. How does God's plan for humans and sharks require them to have almost identical embryos? Tim Barra, doesn't make sense. This is not explained by creation. Jerry Cohen making those sorts of arguments. We've seen those. And the one new one that I have to add here is John Avis. John Avis is a professor at University of California at Irvine, or at least he was at the time of this writing. And he wrote a book about why, I think it was called Darwin and the Genome or something like that. What's that called? Inside the Human Genome. But he made the argument there's just the human genome is just full of problems. It's just got so many problems. There's just no way that this could have evolved. And it's really an appeal kind of the problem of evil, because all these problems are creating all these diseases. And he refers to the embryonic development as well, and says, if you're going to say that this is created by God, that's blasphemous. And he uses that word. This would be blasphemous. So he's, I think he is a Christian, and he's arguing for evolution very, very strongly on that. So that's it. And then my summary is we've seen 3 steps: powerful claims of evidence, very powerful failures of these claims, and in fact, contradictions to evolutionary theory. You really have to admit that this is a huge problem. If you're going to talk about embryology and the embryonic evidences, you have to conclude this is a major problem for evolutionary theory. And then the underlying power, the persistence, because what this really is about is these theological claims. So that.
Yeah. So once again, we see that the powerful evidence for evolution is theological in nature, not scientific. So it fails on the science, and then these theological arguments are used to bolster it. Thank you so much.
And just to fill it in, Rebecca, it's not atheism, which is so often what skeptics of evolution, they want to say it's atheism. And there's no arguments for atheism here. In fact, most of these guys are Christians. There's some atheists, I'm sure, in the group, but the arguments are theological. They're about the doctrines of God. Yeah. They entail doctrines of God.
Now, we have a lot of good comments. I want to start with a set of comments that is actually unrelated to this, but these comments are making a claim, and I have to address this, and I'll say what I want to say about it, and then I'll pass it to you if you want to say something about it. TP Seeker says, ingredients for life have been discovered on 2 different asteroids. There are 20 amino acids that create the proteins required for life on our planet, and scientists have found exactly 14 of them on an asteroid millions of miles away named Bennu. Reuters, May 21st, 2023, 2 organic compounds essential for living organisms have been found in samples retrieved from the asteroid Ryugu. I don't know if I'm pronouncing that correctly, but I just want to respond to this, because when you say ingredients for life have been discovered on 2 asteroids, let me just tell you why I'm objecting to ingredients. Because ingredients implies that you can take things from off the shelf, mix them together, and you'll get something good to come out of it, right? And let me just tell you what they found on this asteroid. You can take it, you can slice it, you can dice it, you can chop it, you can mix it, you can bake it, and you're not going to get life. So what these are, when you're talking about amino acids, right? This is the basic building blocks of a component of life, right? But it is, first of all, amino acids, nowhere close to life. If I gave you all those amino acids right now, I gave them to the best scientists in the world, they cannot put them together and make life. Okay, so to think that this is anywhere close to life, or like some sort of evidence of life in another place or development, it is that, because I think that is the claim that you're making, that this is somehow showing that, we have evidence of the building blocks of life somewhere, and that is just false. And I'll let Dr. Hunter explain, say more about that.
It was 14, so it wasn't the ingredients. Life doesn't use 14 amino acids, it uses 22. So problem right there, right?
And let's say this is a racemic mixture of amino acids. Okay, in life you only have left-handed amino acids. Okay, so this that they're finding everywhere that's not life, you find a mixture of left-handed and right-handed amino acids. Okay, so in order to get life, you would need all left-handed amino acids, and then you've got to get them lined up in a sequence, of a couple hundred to get a basic protein, and a basic protein is nowhere near close to life. Okay, so this is just, it's crazy to think that, and like put this out there as if, see, we've got all the ingredients of life. No. Mm-mm.
Let me add one other thing. This is an interesting, there's a lot of moving parts here, but I would not be too surprised if we found life or strong evidences of life out there, in the ET world, right? So in fact, it already has been found. There has been some pretty good evidences of life out there, but life is very different than 14 amino acids, 14 types of amino acids, as Rebecca, you just pointed out. So on the one hand, it's a joke. Like this is, I was kind of tongue in cheek when I said 14. I mean, you find a few amino acids, you find a population of amino acids, you find all 20, 22, it doesn't matter. That's not life. The jump to actually producing life, you've been kind of brainwashed by the textbooks and so forth that, the origin of life just comes about. You just take some amino acids and it's going to produce life. That's an unscientific, religiously driven idea. It's not a scientifically driven, scientifically informed idea. In fact, just think of it this way. If you took a colony of bacteria and let's say you put it in boiling water or some other caustic environment that broke them up and killed them all, and so you would have all the ingredients of life. So that would truly be, as Rebecca was just mentioning, this would be a case where you actually do have the ingredients of life. So let's say you took that pot and cooled it down and then dumped it out into a lake or something, and would you think life would emerge in a few days or weeks or months or years? Well, no, it wouldn't. So it's not gonna happen like that. But I just want to mention at the other extreme, I think you could actually find life, real life, like microbes or multicellular life or something in the solar system or outside the solar system. I don't think that's something that's too far out.
Now, you said that we have evidence of that. Like, you think pretty good evidence of that. Are we talking like alien evidence? What do you mean?
I think it's more on the microbe level, not on
Okay.
the alien, not on the little green man evidence. It's not something. It's not particularly an interest of mine. I haven't followed it very closely, but I know people who do, that I trust their judgment, and so I've looked at it a little bit.
Cool. Thanks for sharing. Okay, so Mavis says there have been many new discoveries updating Darwin's theory of evolution in the last 167 years. So again, the argument is, why are we talking about Darwin?
So we weren't. So I covered epigenetics. That's 1990s up to today. That's a very new finding. And in the past 10 years, a lot of new experimental techniques, very powerful experimental techniques have been developed to look at how epigenetics works in the embryonic development pathways. So these are major problems. These refute evolutionary theory. They contradict evolutionary theory. It's a big problem for evolution. So if Darwin's theory has been updated, well, let's hear, how has that been updated? How's that working for you? Because everything that has been done has failed. There's been the modern synthesis of the mid 20th century that failed. Now they're trying to figure out a version 3.0 that they can come up with, and they can't come up with one because nothing works. It's falling apart. So what has advanced since Darwin 167 years ago is science. And so we have a lot more scientific information. So you can no longer play word games and thought experiments and rhetorical shell games like Darwin did. Instead, you have to actually own up to the evidence. We're looking under the hood now, and it's not working. What Darwin was saying has fallen apart. So no, it's not working, and there are no new. There's no there. Where's the meat? Where's the? Oh, it's over here. You just didn't look in the right place. There's this new researcher over somewhere who's updated the theory. The theory now accounts for all that. No, it doesn't.
Yep. Okay, so this is interesting. This is I love this because we are seeing the religious arguments. So TP Seeker says, what is the purpose of menstrual cycles? What is the purpose of millions of sperm being ejaculated for one sperm out of millions to fertilize an egg?
But Darwin bemoaned the waste of pollen. Pollen is coming off of our plants, and of course we get hay fever or sneezes from it. But there's just pollen all over the place, and most of it just gets wasted. So you have this waste. And how is that an argument for evolution? Oh, it's not an argument for evolution. That doesn't help evolution. What it does is it's an argument against divine creation. It's an argument against design, against the idea that God created this world. Why would God create these things? So that is a very powerful argument if your theological doctrines don't allow for that sort of a God, right? So this individual, as well as others, obviously have strong theological doctrines. Like, you wouldn't be making this comment if you weren't theologically driven. You have a sense of what God would and wouldn't do, kind of like an ethics. You have this sense this isn't right. Like, God's, if God were to create this wouldn't be right. And so you have kind of this sense of what's right and wrong, and what God would and wouldn't do, or should and shouldn't do. And more power to you. That's what the Bible says. One of the magical things that happen in Genesis is that Adam and Eve learned good and evil. That was a profound verse there in Genesis 3. They now understand good and evil. That was a profound change that came about. We all know that. Everyone here, everyone listening to this has this sense of good and evil. And that informs our thinking. And that doesn't come from evolution. And so you're displaying this now. You have these religious ideas. You have this religious sense that is driving your thinking. And so the first message I have for you and for others is, don't fool yourself into thinking that you're making a scientific argument. Maybe you're not, but I know some people are. They think, oh, I'm going to make this clever scientific argument here. So it's a good argument, and you can think about it and work on it, but don't think for a moment that it's scientific, even though you're using scientific terms like sperm and eggs and fertilizing and so forth. The argument is a theological argument. So you're making a theological argument, which is fine. People make theological arguments all the time, but it's theological. Don't be fooled, and don't fool yourself into thinking that you're doing science. You're not doing science. You're doing religion when you make these arguments. Okay, now once we understand that, then we can go on to saying, okay, let's look at the theology and do we agree with it or disagree with it? And what are the doctrines of God that are informing your view? And are they reasonable? And do they make sense? And are you confident of them? How confident are you? Do you really know God? I mean, how confident would you be going up against God in a debate? Would you feel confident? And, doing things? Well, that's what happened in the book of Job. And so Job was confronted with God. And it's a very actually scary scene in Job 37, 8, and 9. It's a frightening passage to read through. And you read that, I would suggest you go read that. Go read Job 37, 38, 39, 40, and get a sense of what it's like. I think Job felt about this tall when he was going up against God. And God, really put him in his place, to be blunt, And so it's very easy for people to presume upon God and to presume that they know the nature and doctrines of God, know the mind of God. I know if that's what you think God would do, well, that you're crazy, and my God wouldn't do that. Okay, where'd you find that out? So we need to. I guess my second message then is, okay. So my first message was you're making a religious argument. My second message is be careful and think about this a little bit. When you're making religious arguments, you're presuming a lot, and we are finite and sinful. God is not sinful and not finite. So there's a pretty big gap. I'm not saying there's an infinite gap. Because God says, "Come reason with me. Let us come, let us reason." So He is, hoping and enticing that we can, reason together. But He also says, "I am higher than you, and my ways are higher than your ways."
And so I think a key is also. I'm sorry. Did you have more you wanted to say?
Go ahead.
The key is also that. I think God is okay with being challenged. There's people that challenge that are commended or answered or, and so it's okay to challenge God. But I would say take this to God rather than taking it against God. Take it to God. So, God, why did you make the menstrual cycle? Like, that's weird.
Yeah.
God, why all the waste of sperm? That's weird. Yeah. Take it to God. And I've done that with God with many things. And I love some of the answers that God has given me when I take those kind of things to Him. So I encourage you to do that.
Because Rebecca and I probably are not qualified to answer these questions. These are hard questions. But one of the guys that took it to God, his name was Abraham. And he took it to God in a big way. And he questioned Him boldly because God was going to destroy a city because there was evil people in the city. And Abraham thought, gee, that's kind of harsh. What if there's only 50 people in the city, that are terrible? Are you still going to do it? And then he went, what if there's only 45? What if there's only 40? And so it's a kind of humorous but really interesting passage where, I think what you said is right, Rebecca. God doesn't, react necessarily, badly when He's questioned.
Right. Yep. Okay. So Welshmall says optimized assumes an end goal, which is wrong in evolutionary terms. Now, this is in response to you saying that
perfectly adapted for the conditions which they found.
Right.
Yeah. And so that whole idea was, as I mentioned, it derives back to Cuvier. And Cuvier certainly was not an evolutionist. If he was alive today, he probably would be a design guy or a creationist or something. So, I agree with the comment here that from an evolutionary perspective, they're not optimized. And that's not, it's wrong in evolutionary terms. But I think what you may be missing is the crucial premise of the argument is the the in the premise that if God had created the world, it would be that way. So Darwin and the evolutionists are disagreeing with Cuvier in the sense that things are perfectly adapted. Darwin is pointing out, no, things are not adapted. But he's agreeing with Cuvier in that if God were to have created the world, they would be perfectly adapted. Darwin definitely agrees with Cuvier, strongly agrees with Cuvier in the conditional there. That, if God were to have created the world. By the way, I want to make another point there. He assumes an end goal. I agree with you that in principle, in theory, evolutionary theory rejects teleology, and therefore rejects final causes and rejects end goals. But in practice, it incorporates them all the time. So the teleological language and final causes and Aristotelianism runs all through the literature, even to the point, it's not just like an aside, it's even to the point of informing the science itself, the evolutionary mechanisms themselves. And so that's where I think this points out that evolutionary theory is not tenable. It doesn't work. It doesn't work as stated. So the brochure that you got is invalid. The textbook, what Mr. Miller taught you in 7th grade about evolution, it's not true. It doesn't work. So the in theory part, it doesn't work. The way that it actually is put into practice is teleological. And I know that may sound weird, but that's if you look at my channel, you'll see that I've done a lot of videos on that, and I'm actually working on a paper to flesh that out.
Cool. Hey, hello to Wes. Good to see you. Wes has a blog called Creation Questions. It's really excellent. I highly recommend it. Now, Mavis responded and said, "You're special. God answers you." Well, yes, God does answer me. And I believe that he is open to answering everybody who comes to him. The Lord says, whoever calls on me, like, seek me and you'll find, knock and the door will be opened. And so, I am special. Yeah. But God is, you are also special to God. And a lot of people, they have tried to hear from God, and maybe that's you, and become frustrated. And I just want to suggest that maybe the way that you're expecting God to answer is maybe what you actually need to do is learn how to hear how God speaks. Because I've never heard a voice come from heaven. So a lot of people are expecting to hear an audible voice. And I've never heard an audible voice of God. So for me, learning how to hear when God is speaking I would say it's a years-long process, and it's still ongoing. So, but there are ways that God speaks, and we can have a longer conversation about that. Do you want to say something about it, Dr.?
Yeah, I would just pitch in that, similar to you, I've never heard an audible voice either from God. But yet I have no doubt in my mind that he has spoken to me many times and informed me of a lot of things and pointed me in the right direction a lot of times. And so I have benefited, hugely. I have no doubt, of course, the huge benefit that I have no doubt about is that Jesus is alive right now, and without him, we have no hope. So I have no doubt about that at all. And that I believe in Jesus. I believe he existed. He died for us on the cross, and he rose from the dead. I have no doubt. I'm sure of all that. And so the way that God has communicated to me is it's very natural. Like I may have learned something. I may be able to fool myself or have a misconception that I learned something, but it's become apparent to me that I was pointed in the right direction. And so it's kind of just like anyone would learn something, you pick up a book, you read, you learn something, but I've learned things that I was pointed to.
Yeah. And like you were saying, like you have no doubt that Jesus died on the cross and rose again. And really, if someone hasn't heard God speak in another way, please know that that is the way that God has ultimately spoken to the whole world. Is, in the Bible, it even says God spoke to us in the past in many ways. He spoke to our forefathers in many ways, but in these last days, he has spoken to us by his son, who is the radiance of God's glory and the exact representation of his being. So, if you want to see who God is, what God looks like, what God acts like, look at Jesus, who he is, and what he was willing to do for you. Because God demonstrated, he showed you that you're special. He showed you his love for you in that while you were a sinner, Christ died on the cross for you. So please consider that as, God speaking. He has spoken.
Yeah, good point.
Okay. So Stephen says, even if evolution was to somehow be falsified, it is a false dichotomy that creationism would be correct by default.
Yeah, and that's a great point, that we don't want to have this black, white, it's one or the other view, which is how evolutionists argue that creationism is false, therefore evolution is true. So yeah, who knows, exactly how things worked out in the origins, the history of origins, the origins of the world. But I think what's important to understand is that evolution is not this scientific fact that we've been presented with in the schools and in the media.
Yeah, exactly. Okay. Mark says it is not the key evidence, but is still a good evidence that you're lying, that it failed. So I think he's talking about the embryology, and so basically saying that no, it's not true. It hasn't failed.
Yeah, I kind of sense that he must be talking about embryology. And so you're lying part is a giveaway that this is a charged comment. And it's not a dispassionate scientific, I'm looking at the science here sort of comment. There's also a dead giveaway because there's so much scientific refutation and the failure of this evidence. So if you want to say it's not the key evidence, that's fine. It is often cited as one of the pillars, one of the evidences, but there are also offerings. There are books and so forth that do not mention or highly play up the embryological evidence. So it's true. I wouldn't place it as right at the top as the others, but it is often mentioned. It is often claimed to be powerful evidence for evolution. But you're saying it's still good evidence for. So if you try and think through why you believe it's good evidence. What is your argument? Put together the argument. Why is it good evidence? And you're probably making religious steps somewhere in that argument. It's not just an empirical argument, because if you were just looking at the science, then you would have all those problems that I stepped through in this podcast, the epigenetics and the non-homologous development pathways and the PGCs. Those would hit you. Those would hit you pretty hard. And if you were just a theory neutral guy and objectively looking at the empirical science, you would be, wow, how do these evidences really contradict evolutionary theory in a big way. These are serious problems for evolutionary theory. And then if you look at the evidences that are set, I don't know which evidences you're referring to when you say it's still good, but we do have a century plus of literature of evolutionists who have thought through this, obviously, and have presented the best arguments for evolution from embryology. and they fail dramatically. So I don't know if you got something that hasn't been published, you got something else. I don't see how you could say it's still good evidence. But again, that you're lying. So I don't think this represents a thought out, informed, empirical, dispassionate, theory neutral comment. I think this is a charged comment, a partisan charged comment.
Right. But, also in fairness, these are the things that people are being taught. I mean, this is the reason that somebody could jump to the conclusion that you're lying is because embryology is still being taught as this like knockdown evidence for evolution. So basically what you're saying is contradicting probably the majority of authorities that this person listens to.
True.
So it's, yeah, I just, please do critical thinking, consider that people have been wrong, even very smart people, even very educated people have been wrong. And so, right now, this is the situation that this paradigm is being taught, and it's just false. Iron Charioteer says morphology alone proves evolution. The genetics are just icing on the cake, or vice versa.
Yeah, so that is a very heroic claim. Morphology alone proves evolution. You would have to have buried into that claim. There's some implicit cards, that you're not showing here. It's just a comment, so I'm not blaming you for not giving all the details, but there's a lot poured into that phrase when you say morphology alone proves evolution. Of course, I know what the arguments are because I know the literature, and so I know what the arguments are that morphology alone proves evolution, and they are theologically laden. They are contingent on theological claims about what God would and wouldn't do, the doctrines of God. You cannot make that claim from the empirical science. That is a ridiculous claim just from the perspective of the empirical science. I'm not saying you're ridiculous. I'm not saying the comment is ridiculous, because I'm assuming that you are making a claim that is reflective of the literature, is typical in the literature. It's traditionally been made for a century or more. And so yes, there are strong arguments, and that's why, Rebecca, I always say, it's a strong argument. You say, well, why is it strong? And these arguments are strong, and this comment kind of reflects that. This is why this is such a strong theory, and the evidences are so strong and so powerful, because look at what it's rendering here. People making these sorts of comments. Morphology alone proves evolution. Genetics is just an additional layer on top of that. Wow, like, how could you possibly say that in light of the empirical science? So obviously there's theology at work here, theology that you're probably unaware of.
Yes. God is nowhere, or God is now here? I leave it to you to figure it out. And says genes being turned off has a different meaning when considering randomness in evolution theory.
Genes being turned off has a different meaning when considering randomness in evolution theory. It doesn't help.
Okay, too hard. Okay, good to see you. God is now here. Look, guys, there are so many still good comments remaining. So we've almost been going 2.5 hours.
I'll try not to say I have long answers here.
So Mike, good to see you. And Reezy, good to see you. Thanks for all your comments. So, okay, we can stop or we can keep going. It's up to you.
I'm good for a little bit more. Let's keep on going.
Okay. So David says natural selection doesn't select on gene level, only on an organism level. Therefore, it can't select positive mutations without selecting the negative ones.
Okay, interesting point.
Okay.
Not hugely relevant to this podcast.
Okay. And let me see. So Mavis says, what about the evidence for your God? Do you apply the same scrutiny for evidence for your God?
Mm-hmm. Yeah, that's a good one. First of all, I would say yes. The evidence for Jesus is, from a theory neutral, a religiously neutral perspective, very persuasive and compelling. You would have to come up with some very creative, heroic explanations to get around Jesus, even his resurrection. But I understand how that is a heroic claim, that somebody rose from the dead. But I'm also not claiming that that it's a scientific finding and that it is a religiously free, neutral, theologically free finding. Like, my concern about when you say, do you apply the same scrutiny? I think I do, but I want to make clear that evolutionists are claiming that it is a fact, and that fact of evolution comes from empirical science, theory neutral empirical science, without any theological premises. That's just a false claim. Both the evidence doesn't make the case, and the theory is full of theological claims. So I want to make that clear, that what evolutionists are claiming is false. They're misrepresenting the theory. And I realize that they may be misled themselves. It's deceptive. It's a huge deception. So I'm not meaning to call them liars or anything like that, but they are misrepresenting the theory. So when you kind of equate, evolution with, my religious beliefs, okay, I just want to make clear, when I say I know that Jesus is risen, Jesus was a real person. He saved us. He saved me, and he's alive. And without him, we have no hope. I know all these things to be true, but I'm giving you a witness. So it's like if you go to a court of law, and you're the jury, and there's this guy he's on the stand. I bring forth Cornelius here, and he says a bunch of stuff. Okay, you take him as a witness. You take that, and you weigh that. Is he telling the truth or what? So I'm just giving you a witness. Now, I could get into arguments about why I think it's true and so forth, but that's my witness. That's my experience. And so you take that for what it's worth. I'm telling you, I don't have any question about it. I know that Jesus saved me, and I know that Jesus is the Savior, and no one goes to heaven except through Jesus. It's not like there's a bunch of Jesuses, or there's there's this other guy and there's this other guy. Jesus is the way, the truth, and the life. I know that to be true. If you want to, so that's my witness to you.
Thank you. And I like what anarcho-republican says God speaks to me explicitly in scripture. The Sermon on the Mount is in black and white and red. I like that comment. That's right. If you have trouble hearing God's voice in other ways, you can always open up the Bible and just read straight and see what God is saying to you. So
actually, I think that's a really good comment. That's a good reminder. Thank you for that comment, because that's probably the first answer you should give to somebody, right? If somebody says, well, God doesn't speak to me. Well, have you ever read the Bible? Because God will speak to you big time. Right. And that's why I tell people, a lot of people have never read the Bible. It's like, it's the most influential book ever written. I mean, at least crack the Bible. Start with the book of Matthew and just read one page, half a page per day.
Yeah.
It's 15 minutes, and just do that every day. Have a habit every day, and in 6 months, you'll finish the New Testament. God will speak to you,
and at the very least, you'll be a more educated person.
Yeah.
Because if you haven't read the most influential book of all time, you're missing something.
Yeah, it's time well spent. You will be culturally educated.
Right. Okay. Well, thank you everybody so much. I want to finish just with a last comment from Mehrashad. If you guys don't know, Mehrashad is in Iran, and he's a very good, supporter of the channel, but hasn't been able to support much lately because of internet out and everything. But thank you, Mehrashad. Glad that you are doing well. Mehrashad says, I'm eager for it. Developmental biology is a pretty difficult but marvelous branch of biology.
Fantastic. Thank you for the comment, and best wishes to you in these trying times. I'm sorry for what's going on.
Yes, and I am praying for you, Mehrashad. Thank you so much, Dr. Hunter. Appreciate it.
Thank you, Rebecca.
Thanks, everybody, for watching.